Hi guys, this is Liz and I'm going to be talking about biofilms today. What is a biofilm? So a biofilm is a layer of bacteria or other microbes that stick to and grow on the surface of a structure. That structure can be household items, industrial pipes, contact lenses, medical devices such as catheters, and then plant and animal tissues. So the development of biofilms involves 6 steps. The first step being free living microbes to be living on a surface. And like I said, those surfaces can be contact lenses, catheters, teeth, plastic tubing. The second step senses the microbes on that surface using fimbriae and are triggered for microbes to attach. The surface then develops a gooey substance called extracellular polymeric substance, or EPS for short. So the third step involves EPS to adhere cells to one another and create microenvironments within the biofilm that require nutrients and then water channels. The biofilms form as a result of quorum sensing, which I'll talk about in a bit. That triggers the cells to change their shapes and their biochemistry. And in the fifth step, it involves new cells arriving to the biofilm, which could include new species or new water channels to be formed. And then the sixth and final step, which is kind of shown in 5 on this picture, is microbes escaping the current biofilm they are currently living in to go back to being free living microbes. I think it's interesting because I touched on where biofilms are mostly seen, but I think it's interesting as far as they're also found on rocks and stream beds because I never really knew that when I was walking in like a river, a lake, to have feel those rocks and feel that kind of gooey substance on those rocks. So I thought that was interesting. So kind of touching on quorum sensing a little bit more. I really liked this picture from the book and it kind of was easy to look at it and kind of understand it a little bit more. So quorum sensing is defined as a microorganism's response to the density of nearby microorganisms. Microbes are able to secrete the quorum sensing molecules, as you can see from the red triangles, and then that can help communicate to the number and the types of cells among that biofilm. So, how these molecules work is by bacteria synthesizing and secreting the signaling molecules, like I said, is the red triangle, and they can be detected by the protein receptor, which is kind of that blue Pac Man. So as you can see from the low density on the left, the signal kind of moves away from the receptors, whereas in high density, that signal is able to reach that receptor. So biofilms can have positive and negative effects on our body. Mostly negative, but there is a couple positive that I have found. So gut microbiome is one, and then the second includes biofilms commensal bacteria that can stop the colonization of pathogenic bacteria through host cell immune defenses and prevention of adhesion. Like I said, there are a lot of negative effects. One being cystic fibrosis. The persistence of biofilms in cystic fibrosis leads to the development of a distinct antibody response. Biofilms can cause chronic infection. Because of that, they show increased tolerance to antibiotics. So, the methods for controlling medical biofilms include 3 different approaches. They're still doing a lot of research to today, but the first approach is obviously inhibiting the initial attachment by kind of re engineering the surfaces that biofilms grow on. The second approach is kind of disrupting that maturation process. And then the third is to interfere with that quorum sensing. And then there's a lot more research on biofilms. They touched on a couple in the book that I found really interesting. So one method includes that drugs block microbial cell receptors, which would disrupt the communication among the cells and inhibit biofilm formation. So I guess that can kind of sense on that signal um, signal interfering, as I touched on kind of before. That is currently successful in mice and it's being considered in humans. Another method being researched is the use of an enzyme to break down that gooey substance, that EPS, and enhance the penetration of those antibiotics. Um, Other than that, I think that's it. So I hope you guys learned a little bit more about biofilms from this. I certainly did. I hope you guys have a good rest of your night.